Welcome to the Diabetes Bio Podcast, a podcast for the Diabetes Journal, the flagship research publication of the American Diabetes Association. Each month, we will interview one or more authors from the feature articles, the papers of the month, or other articles published in that month's issue of the Diabetes Journal. We also have a Sweet Talk segment of the podcast, which is more of an open format discussion of additional articles, conferences, or major events in the field we'd like to highlight. My name is Darleen Sandoval, and I'm joined by my podcast co-host, Dr. Kirk Haberger and Dr. Kevin Williams. How are y'all doing today?
Hey, Dar.
Doing pretty good.
So we are recording this in October, just 10 days away from Halloween. So, Kirk, I'm curious what the kids are dressing up as.
Oh, that's right. That's right. We got let's see, we've got one who's too old, too cool for school, no more dressing up. And then the younger two are going as a banana and
Nice.
I guess a giant banana in this case. And then the classic morph suit, right? The spandex black disappear into the darkness.
Nice.
Yeah. So
I have a friend who used to wear a banana suit running marathons.
oh, that's great. I love the banana suit and also the T-Rex, the inflatable T-Rex, right?
Yep.
Yeah, I've seen that one too. Those are great.
Hopefully the friend washes the banana suit in between races.
Yeah, hopefully. It's how do you get a big banana into the washer? I don't know.
I know.
On the other hand, it gives you more space to run, right? Nobody
That's
else wants
true.
to be near you.
That's true. That's true. Kevin, do
Also
you
the
dress
smell.
up for Halloween or do you participate at all?
Not really dress up, but we usually get a couple containers of candy and wait by the door and greet all the kids as they're coming by and doing their trick-or-treating.
Yeah. Yep. Awesome. All right. Well, we should actually get some work done on today, I guess. Kirk, why don't you tell us what we have in stock for our interviews today?
That's right. So for November, we're going to be joined by lead author, Dr. Nicholas Maxwell, and corresponding author, Dr. Claudia Grillo, for a discussion on their article titled Leptin Activation of Dorsal Raphe Neurons Inhibits Feeding Behavior. Then we get the opportunity to chat with Dr. Marcus Fruttiger, professor of ophthalmology at the University College London, at the Institute of Ophthalmology there, about his paper, Deciphering the Connection Between Microvascular Damage and Neurodegeneration in Early Diabetic Retinopathy.
And Kevin, we have a fun Sweet Talk segment coming up. So why don't you tell us about that?
Yeah, this is a really special one in honor of World Diabetes Day, which is November 14th, and National Diabetes Month in the United States. We're speaking with Dr. Giles Yeo, professor of molecular neuroendocrinology at the Medical Research Council Metabolic Disease Unit and scientific director of the Genomics Transcriptomics Core at the University of Cambridge. We'll be discussing his insights on the intersection of obesity and diabetes, how genetics influences these conditions, and what that means for treatment and prevention. The conversation includes active research, but also touches on topics of general interest. So whether you're a scientist or someone curious about how our genes affect our metabolism, you'll want to stick around.
Awesome. Let's jump in.
Now, I want to alert you to a special offer. The American Diabetes Association is extending a special offer for new and renewing members. When you sign up for a professional membership to the ADA, use the code FALL24, that's FALL24, during checkout. And this will get you 15% off the yearly membership price. And if you sign up for a 2-year membership, you'll receive a 15% discount and a free It Takes All of Us t-shirt from the ADA. In addition, you will get exclusive discounts on ADA's scientific sessions and other meetings, a subscription to one of ADA's journals, a copy of ADA's Standard of Care in Diabetes, and other membership exclusive discounts. Again, that's 15% off a 1 or 2-year ADA membership using code FALL24 in all caps, that's FALL24 at checkout. And the offer expires November 30th, 2024. Don't miss out on this fantastic opportunity to expand your commitment in the fight to end diabetes by joining the American Diabetes Association. 。
For our first paper of our November podcast, we're going to discuss a feature article entitled Leptin Activation of Dorsal Raphe Neurons Inhibits Feeding Behavior. We are joined by lead author Dr. Nicholas Maxwell and corresponding author Dr. Claudia Grillo. Dr. Grillo is a research associate professor in the Department of Pharmacology, Physiology, and Neuroscience at the University of Southern Carolina School of Medicine in Columbia. Dr. Maxwell is a former student of Dr. Grillo and is currently a postdoctoral fellow at Duke University School of Medicine. Welcome, Drs. Maxwell and Grillo. Congratulations on your paper, and thank you so much for being a part of this month's podcast.
Thank you for inviting us.
Yes, thank you very much. Glad to be here.
Now, I didn't send you this question, but as an icebreaker, I'm curious if there's any part of your personal life that informs or highlights the work that you do in the lab. And we'll actually start with Dr. Maxwell on this one.
Any part of my personal life that highlights the work in my lab.
Or that intersects with it, right? I mean, is there a crossover there? Of course.
Yeah, I mean, absolutely. I mean, studying food intake and feeding behavior. I love to eat. And so, I'm a
with
big
you.
food junkie, and recently moving to Durham, North Carolina, there's a lot of good food to try. With loving to eat, there's also a bit of controlling how much I eat. So that's also an important part of it. So I do try to exercise and eat healthy when I can, but I do love good food. So
Awesome.
good.
Who doesn't?
That's right. And Dr. Grillo?
Oh, this is a difficult question. Well, maybe this is not so related to food intake, but very related to research in biomedical sciences. When I was a little kid, my parents bought for me as a Christmas gift a chemistry set. And that was the best toy that I ever had. I think that I was around 8 or 9 years old, and I was playing with this set all the time in my mother's kitchen, having my grandma as an assistant. And since then, I have been in the lab. I started in a kitchen, and that's why maybe I finished in food intake.
There you go.
That's great. Claudia, first we'd like to give our authors the opportunity to highlight any other contributing authors or collaborators to this work. Do you have anyone you'd like to acknowledge?
I would like to acknowledge everyone. I will start acknowledging Dr. Steve Wilson, who designed the viral tools that we use for this manuscript. Also, Dr. Feidel, who is the expert in microdialysis. That is an important component of the results of this manuscript. And also my long-term colleague, Dr. Regan, who was always very generous, supporting us with all the resources from his laboratory. And I think that maybe Nick has more people that he wants to acknowledge.
Yeah, absolutely. So there's a lot of people who contributed to the work of this project, in addition to the faculty members that Claudia just mentioned. But two I want to highlight first is Arcora Smiley and Victoria Macht, both of which were previous students with Dr. Grillo in the lab. And they really helped her to sort of lay the foundation for this project, do a lot of those early pilot experiments to show the validity of the work that we were doing and the direction we were going. A lot of the work they did was really foundational for the work that came after, and a lot of the work that I've done. And then throughout my time there, I worked with quite a few technicians who were invaluable extra hands in the lab, and they were also very brilliant in their own ways in contributing to this project. And that's Alia Sadek, Francis Loyo Rosado, Daniel Giles, and Jennifer Woodruff. And then we had 2 undergraduate students on this paper who helped so much, Tori Glass and Don Zell. It was a delight to have them and work with them for these studies.
Awesome. Science is definitely a team sport. At least the best science.
Yes,
that's right.
All right. Now, Claudia, can you give us a little bit of an overview on your research program and how this project came about based on that overview?
Okay. I was always very interested in appetite, in the control of appetite, maybe because this is an essential thing for life and is critical for animal survival. But I didn't start to study appetite until I have my independent laboratory. But my background was in diabetes and obesity. And, we were always working with hormones like insulin or leptin and the effects of these hormones in the central nervous system. I was very interested in the effect of these hormones on mood disorders like depression. And depression is very, very related to food intake. When you have a diagnosis of depression, probably that is accompanied by changes in food intake. It could be increases or decreases, changes in patterns or changes in the preferences, but it's always there. And then we have a nice link to study between depression and food intake. And then I was thinking, okay, maybe there is an interaction between leptin and serotonin that is one of those neurotransmitters that are very well known by being altered in depression. But in addition to be in the depression field, serotonin is also in the regulation of food intake, and say, okay, probably there is a good interaction between leptin and serotonin. And I decided to study this. And when I started to look in the literature, I found other groups that were investigating this, but there were a lot of discrepancies in the field. I said, oh, this could be a problem or no. Maybe this is a good problem to have. There is many more things to do here. And then I started to look at the interaction between leptin and serotonin. And the NSF was very interested in my proposal, and it was funded. And okay, we developed this research, and it was great for Nick because he was able to have his thesis on this subject.
Awesome.
That's great. So I want to dig in a little bit more to the background here, right? And specifically in this article. So it seems like there may be a little bit of discrepancy, and you mentioned that about the role of serotonin and the 5-HT receptor and leptin. Can you tell us a little bit about the overlap there and what took you to the Raphe nucleus there?
So you're absolutely right. There are some discrepancies, and so starting out, there are lots and lots of studies that establish a very clear link between the neurotransmitter serotonin and appetite. And these studies, as Claudia mentioned, but these studies range all the way back to early pharmacological studies done in the mid 1970s and through the 80s, and those continued on and sort of developed as the techniques developed to continue to show that serotonin has an effect on food intake. Eventually, once leptin was discovered, they started to look a little closer at where leptin works in the brain and how it may work. And so the hypothalamus was the easy first target. And so a lot of studies really focused on the hypothalamus, but there were some studies that showed that there were leptin receptors expressed throughout the brain, including in the dorsal raphe. And so once those studies had come out, some other labs started to also look into this. In 2011, there were two different studies that came out from different labs. One study, the authors didn't find any overlap between serotonin raphe neurons and neurons that express leptin receptors in the raphe. There were no electrophysiological changes after the introduction of leptin into the raphe or onto raphe slices. This really seemed to show that there's really no interaction of leptin with the dorsal raphe and that it really has no effect on food intake through this pathway. But there was another paper that also looked at the interactions between leptin and the serotonin system. And they found that genetic inhibition of serotonin production reduces food intake. But when the leptin receptors are removed from only serotonergic neurons, this reduction in food intake is abolished. And so that study seemed to show that there is some sort of interaction between the leptin system and serotonin. So with both of these papers kind of coming out around the same time, it sort of split the field into people who, you know, decided that leptin and serotonin were not involved with each other and that they're not really influencing appetite in any way. And then others who weren't so sure. We saw this discrepancy, and I think, and I say we, but Dr. Grillo was the one who was pioneering this work early on. But she really identified that there's room for some clarity here. And so that's what really spurred all of these projects and all of these studies to really sort of clear up what's happening here and whether or not leptin and serotonin are interacting.
Awesome. So I'll stay with you, Nicholas, and I want you to tell me about some of the, one of the complications of using optogenetics and chemogenetic strategies is that it is kind of hard to figure out how to validate that you're doing what you think you're doing to the neurons, right? Can you tell us a little bit about what you did to give yourself confidence that you were activating those leptin receptor neurons in the dorsal raphe?
Yeah, and Claudia may be a little bit better equipped to answer this question, but
Feel free to jump in if you want.
Okay. Okay. Yes, we were very satisfied with the pharmacological approach, but we understand that maybe as there is a discrepancies in the field, it would be better to confirm using other techniques. And optogenetics sounded like, okay, that could be a good way to evaluate this. We didn't have any experience with that, and I always like to introduce new techniques in my lab, and then I said, okay, let's try the optogenetics. And there was a lot of information and a lot of good companies providing good resources. And then we started to work with some people who are experts in designing viral tools. That was Dr. Stephen Wilson, and with him we designed a lentivirus that has that express the channel rhodopsin, but under a promoter for leptin receptor, so that we were targeting just the cells or the neurons that express leptin receptor. And it was a little challenging at the beginning because it was a completely different field for us. But we had a lot of experience with stereotaxic. That was not a problem. The problem was establishing the best conditions for the optogenetics. But we did a lot of troubleshooting until we start to see changes in food intake when we stimulated the channel rhodopsin expressed in the arc weight. And there was no doubt that the arc weight regulate food intake. And then that was the first target that we used. Okay, and then when we had the conditions for the arc weight, we saw a good decrease in food intake. We decided, okay, now we can go to the Raphe and repeat the experiments, and we have good results. And something that is important, you said about the controls, no? It's like how we felt like, okay, with this technique. Something important was for us to have the negative controls, and then we have a virus that did not express the channel rhodopsin. And we did all the experiments, the surgeries, injecting the virus, the stimulation with the blue light, and also with the yellow light as a negative control, and even the sham. And we didn't see any changes due to all these manipulations, no changes in food intake. Okay, and then we felt the control is working very well. And then what we are seeing with the blue light is real. And
Yeah.
then when we went to the Raphe, we observed a similar decrease in food intake that, of course, in magnitude was not as big as what we saw in the arc weight. But one of the benefits of the optogenetics is it allows us to study circuits. And then we were able to inject the virus into the Raphe, but instead of stimulating the cell body, we stimulated the terminals that reach the arc weight. And then when we stimulated those terminals, we saw a decrease in food intake. And then, wow, we were so happy to confirm that the circuit was functional.
That's very impressive, and it's nice to see all those controls, right?
Yeah.
Yes,
Yeah.
yes.
So I have to say, I want to highlight the figure you have where you do the administration of leptin into the Raphe, and then you do the microdialysis measurement of serotonin in the arcuate, and then go back and pharmacologically inhibit, right, serotonin signaling. Really, really cool study, right? Remind me, because I'm not a brain guy, how accessible is the Raphe to leptin, right? How does that signal, or sorry, how does that hormone activate those neurons, and what proportion of the leptin effect to suppress food intake is actually coming from the Raphe?
Okay, we consider that Raphe is a contributor to the control of food intake, but the main command is in the hypothalamus. So far, it's like nobody has demonstrated otherwise. But going back to your question, we did some controls in which we injected the leptin into the Raphe, and we look at the phosphorylation of STAT3 that is in the pathway of leptin, is considered a marker for leptin function. And we studied the expression of PSTAT3 in different areas of the brain to double check if there was a leak of the hormone to other areas of the brain. And we didn't see in the hypothalamus, because it could be that the injection of the leptin in the Raphe, and the Raphe is very close to the ventricles, and then it could be a leaking to the hypothalamus. We didn't see that. And something that helps to confirm this is the optogenetics, because with the optogenetics, we injected the virus into the Raphe, and we don't see a spread of the virus beyond the Raphe. And later we have to do a second surgery in which we put a fiber optic into the Raphe to administer the light. And the penetrance of the light is very, very small. I don't think that it go beyond 1 millimeter. And then that confirms that everything is happening there compared to the pharmacological experiments in which, okay, I understand that we can think the drug maybe went to other places, but the light can't go to other places as easily as the drug.
Nice.
And again, your confirmatory controls and experiments are great here.
Yeah. Nicholas, do you want to maybe summarize the circuit a little bit? Because we've kind of touched on it in different aspects, and then highlight anything else, any other results that maybe we haven't discussed yet that you'd like to share.
Absolutely. Some of the early experiments that we did with the pharmacology and looking at how leptin reduces food intake was really like the confirmation cornerstone for the rest of the study. But then from there, we wanted to understand exactly where this was affecting. And so, like, yes, the Raphe is being activated by leptin, but then what's the Raphe doing? For us, it was very clear that it was likely that the Raphe was projecting to the hypothalamus. And so we did a couple different experiments in which we injected retrograde tracers into areas of the hypothalamus that are important for food intake. And one of the ones that we really honed in on was the arcuate nucleus, which is the main regulatory hub for feeding behavior. It houses many different types of neurons that control food intake and respond to a variety of stimuli to control food intake. When we inject this retrograde tracer into the arcuate, we see a lot of neurons within the Raphe nucleus that are projecting to that region. Using that as sort of a baseline, we were then able to identify, okay, these Raphe neurons that are projecting to the hypothalamus, are they responding to leptin and are they serotonergic? And so we did this through both pharmacology mixed with immunohistochemistry, as Claudia had described previously, looking at the activation of phosphorylated STAT3 in neurons following leptin administration. And we also used in situ hybridization, or RNA-scope, the proprietary form of it. And we used that to look at the mRNA expression of leptin receptors within the Raphe nucleus. And both methods really showed us that around 9% of neurons that project to the arcuate nucleus are also making serotonin, and they respond to leptin or express leptin receptors. And so that was really, really encouraging to see that, we were on the right track and that, these neurons are existing within the dorsal raphe. And these are likely the ones that are driving a lot of this behavior that we're looking at.
Great. Dr. Grillo, as we finish up here, I wonder if there are any key points or thoughts that you'd like to leave us with, or maybe share some insight into your future work.
We would like to keep investigating in this circuit. In fact, the serotonin receptors that are expressed in the hypothalamus, and that we show that inhibiting one of them, we attenuate the anorectic effect of leptin. We just use an antagonist for one of the receptors. We use for the 2C receptor, but there are so many serotonin receptors and several of them are related to food intake. And then we want to keep looking at those receptors. And we also want to study other neurotransmitters that are also produced in the Raphe, because Raphe is the main source of serotonin, but also produce glutamate and GABA. And we would like to keep looking at the interaction of leptin with other neurotransmitters. But the main point that we can see here is that the Raphe is able to regulate food intake through leptin, and other laboratories have shown that even GLP-1 has an effect on Raphe interacting with serotonin. It's like we are finding new functions for the dorsal Raphe nucleus.
Very cool.
Excellent. Well, Doctors Maxwell and Grillo, we want to thank you for joining us today and sharing your expertise and for discussing your recent work on leptin and serotonin signaling and the regulation of food intake. I encourage the listeners to read the full article in the November issue of Diabetes. Thank you.
Thank you.
For our next article, we're going to be discussing the November paper of the month, titled Deciphering the Connection Between Microvascular Damage and Neurodegeneration in Early Diabetic Retinopathy. We're joined by senior author, Dr. Marcus Fruttiger, professor of experimental ophthalmology at the Institute of Ophthalmology, University College, London. Welcome, Dr. Fruttiger. Congratulations on your paper, and thanks for being a part of this month's podcast.
Thank you for choosing our paper as a paper of the month. We're very honored to being highlighted like this.
So we like to start the conversation with an icebreaker. So what is a fun fact about yourself that has nothing to do with science?
My fun fact on science related.
Yeah.
I'm Swiss. I love skiing, but I barely ski because I live in London.
Well, it's an excuse to go home at least, right?
Yeah, no kidding.
Yeah, there is.
All right, we do like to give our authors the opportunity to highlight any other contributing authors or collaborators to this work. So would you like to start with that, Dr. Fruttiger?
Yes. I would like to highlight Jian Yang, who's the first author of the paper. She was a PhD student in the lab, and the work that's been shown in the paper has formed a significant part of her PhD thesis. It wasn't the only thing she's done in her thesis, but she spent quite a bit of time on this. She has now moved on, is a postdoc in a different lab, but I'm very grateful for her contributions to that. And also, I have to mention my collaborators at Moorfields Eye Hospital, who are clinicians who I very closely work with. And, the people who work in the eye banks, in particular, Mei Dong Song, Sue in the Sydney Eye Bank, and the people from the NHS Blood and Transplant who provide us with the human postmortem tissue, which we used for this study. And obviously, we also have to acknowledge all the anonymous donors and their families who have gifted us the research tissue that was required to do this study.
Yeah, absolutely. And so much of our science comes from teamwork, right?
Absolutely, right? Absolutely. Yeah. We couldn't do it on our own.
So moving on, maybe you can give us sort of an overview of your research program and then how this specific project grew out of that.
Okay, so Initially, I was what people call a basic scientist in my early career, and doing research in complicated transgenic animals and so on. But as I started to work with clinicians at Moorfields Eye Hospital, I became more and more interested in what is actually happening in the human eye, obviously, because our focus is on ophthalmology, on eye diseases, in particular on retinal eye diseases. And that kind of led to a shift in my lab, where I recognized that the insights which we gain from model systems, such as animals or cell culture and so on, they are not always easily translated into human applications. And that is really, really important to learn more about the human disease. In ophthalmology, this is not straightforward because you can't just take a biopsy, right, of the retina. So we do depend on post-mortem analysis, and that again is complicated and takes time, and therefore doesn't give you any options to for experimental manipulation. So it is difficult to research what is actually happening in the human eye. But nevertheless, that is actually the main focus in my lab, where we do try and understand first what's happening in the human, and then if we do find any leads, then follow those up. Secondly, in model systems to then explore mechanistically how things might play out. And the main diseases which we're interested in is the retinal diseases that tend to have vascular complications. So that is diabetic retinopathy in this case, but also age-related macular degeneration and others, less common diseases.
Can you tell us a little bit about retinopathy itself? Maybe a little bit of the basics of the pathology or even the clinical incidence, et cetera?
Yeah, so with regards to diabetic retinopathy, it is a complication of diabetes. Now, people with type 1 diabetes, they have a much higher risk of developing diabetic retinopathy. Type 2 diabetes, still around a quarter of them will go on to develop diabetic retinopathy. And then within the ones that do have diabetic retinopathy, about 5 to 10% then move on to going to severe stage, into a sight-threatening stage that really needs intervention. Problem is that from a pathological point of view is that we don't quite know for sure what's happening here, but there is certainly a link to high glucose, hyperglycemia in the blood that damages the blood vessels, not just in the retina, but actually in many different places in the body. And in the retina, that leads to little problems. Initially, it just starts like with little leakages, little exudates of serum or blood into the retina. Then blood vessels can become abnormal, deformed, these little microaneurysms, and some of them will start dying as the disease progresses. So you start losing blood vessels, and usually it's the smallest ones in the retinal vasculature, so the capillaries that start to die off. Now, once you start losing vascular support, obviously all the tissue that needs the oxygen from these blood vessels is becoming threatened, and some of the cells in the vicinity of that tissue start to die off. And that obviously then has an impact on the retina, and it can lead to further complications where then once the retina detects that it really lacks oxygen, it starts to secrete a factor that stimulates the growth of new blood vessels. But this is very dysregulated, and then too many vessels grow and the whole thing gets even worse. So that's roughly what's happening in diabetic retinopathy.
Thank you.
Great. So, Marcus, one of the very important techniques that you highlight in this manuscript is the evaluation of retinal vascular perfusion. Can you tell us a little bit about the previous techniques to evaluate that endpoint, right? And then what you've utilized in this specific study?
Vascular perfusion is something that happens in living people with a beating heart.
Right.
And in patients, you can quite easily actually, you can measure it, you can visualize it. And there's two methods. One is where you inject fluorescent compound, fluorescein, into the bloodstream of a patient. And then you look into their eyes and you image the back of their eye, and you can see how the retinal vasculature starts to light up with this fluorescent compound that eventually reaches the eye. That is now a little bit out of fashion because there's a more modern technology called angio-OCT. The angio-OCT works similar to ultrasound imaging, but based on light, where you can create an image that creates like a section through the retina, and you can image the eye without injecting any dyes into the patient.
Oh, very cool.
If you image that on a continual basis, the changes in that image, those clever detection methods, actually allow you to see where there's blood flowing. And based on this, is this technique called angio-OCT that allows you to visualize where the blood vessels are and where there's perfusion. Actually, it doesn't really show you where the vessels are, but where there's movement in the retina. Now, that's straightforward in human patients. Now, obviously, if we want to look at that at the an assessment of vascular perfusion in postmortem tissue, that seems like impossible superficially. So what we used here was a trick, basically, and that's based on the fact that on two things, really. That firstly, when blood vessels die anywhere in the body, you lose the cells inside the blood vessel, and more importantly, the internal lining of the blood vessels, the endothelium. And it's impossible for our blood vessels to carry any blood without that endothelium. So the endothelium prevents blood from clotting and so on. And so it's essential to have an intact endothelium. So when blood vessels die and you lose your endothelium, that means the vessel immediately becomes impossible to perfuse that vessel. However, blood vessels, they secrete extracellular matrix sleeve around them. And that is fairly tough, made from collagen and other extracellular matrix molecules. And that tends to persist, that tends not to die off very quickly. And in fact, we've looked at some postmortem tissue where for many, many years, we detected the extracellular matrix outlining the previous vessels, and they persisted for many years. So some people call them ghost vessels because they no longer contain any cells. They're only containing that kind of extracellular kind of sleeve. You then, if you go in histological sections, you can distinguish between these two states. So we can quite easily stain for the collagen in this extracellular matrix sleeve that persists. And then if you simultaneously image for endothelial cells themselves, we can see where the sleeves that contain endothelial cells on the inside and sleeves that no longer have endothelial cells on the inside. And therefore, you can then make an immediate assessment that, well, this profile of a vessel here is perfused because it still has endothelial cells in there and the other one isn't. So admittedly, it's an indirect readout. It's an assumption that if you have endothelial cells, you will have perfusion, but that's our indirect readout. But we certainly can say that if a vascular profile does not contain endothelial cells, it's certainly not going to be perfused.
Got it.
Let's dig a little more into the data that you collected. So you have retinas from 14 donors, 3 were non-diabetics, 10 diabetics without retinopathy, and 1 diabetic patient with retinopathy. And you found some differences with capillary density between these groups. So can you tell us a little bit about those findings and maybe some potential explanations for those findings?
Yes, so initially we were interested in diabetic retinopathy, and as scientists, we're always interested in where disease starts, right? Where does it all start? Because that's ultimately the aim of my lab, to understand these pathobiological mechanisms that drive the disease down this pathway where eventually it leads to very, very severe complications. Because we depend on postmortem tissue, it's actually not that straightforward to find postmortem tissue from people with diabetic retinopathy. And once the diabetic retinopathy is fully established, you're looking at kind of a train wreck, right? There's a lot of damage in the tissue, and it's quite difficult to interpret. So ideally, you pick tissue that is from very, very early stages of diabetic retinopathy. Problem is, how do you find those, right? So what we decided to do is we decided to screen in the first instance just anonymous donors who we knew had diabetes, but not diabetic retinopathy. In those, we just started to sample for these non-perfused blood vessels, because that's an indication of non-perfusion, and that we know that is certainly something that contributes to disease progression in diabetic retinopathy. And we found that in diabetics who have not been diagnosed with diabetic retinopathy at this stage, although it's a little bit difficult because they're anonymous and we didn't have the full clinical records of all these patients, but they didn't come with a label of diabetic retinopathy, so we assumed they were relatively normal. We found that there were still a significant number of these non-perfused blood vessels in cross sections of their retina. And what was particularly intriguing was that it primarily affected blood vessels that were sitting in a deeper layer of the retinal vasculature. So put this in context. So the retinal vasculature is a network of blood vessels that enter through the optic nerve and then spread across the entire retina. Initially, the blood comes in a superficial plexus. It spreads out across the retina in the superficial plexus. And then there are vessels that go down into the retina, into a deep plexus. And then the blood drains back towards the optic nerve, out again of the eye. Now, what we found is that the deeper plexus was particularly affected in these very, very early stages of vascular pathology, which we found in these early stages of diabetic retinopathy, you can call it. So even though it's officially, these people, if they would have gone to a doctor, he may not have diagnosed them as diabetic retinopathy of just looking at the fluorescein angiogram, because the superficial plexus would have looked relatively normal because there was no damage really obvious in the superficial plexus, but it's the deeper plexus that showed some of this capillary dropout. Now, there's quite a few now clinical papers that have used angio-OCT that are imaged in living people, because angio-OCT allows you to distinguish the superficial plexus from the deeper plexus, and they have come up with similar findings. But our study was the first one actually has confirmed in human histology to show that really the deeper plexus within the retina is the one that is affected first. So this has two consequences. Well, this has a consequence. On the one hand, it means that it has been so far overlooked, but it confirms the OCT findings. But it also means that these, in an OCT finding, this readout may have validity as a really early readout for oncoming diabetic retinopathy that is about to develop. Because as I said, these postmortem samples which we analyzed, so these 14 donors, they were not diagnosed with diabetic retinopathy at the time.
Could be a really important finding, right?
Yeah.
Yes,
Yeah.
but so why the deeper plexus is affected, initially, we don't know. So that you could say, well, maybe it's the most peripheral, if you look from a blood circulation point of view, right? So the superficial plexus is closer to the heart, if you want, whereas the deeper plexus is further. But that's pure speculation. So we don't know why the deeper plexus is affected first. It may have something to do also with the cells in the retina that surround the deeper plexus.
So, Marcus, I wanted to ask about your findings on capillary dropout. It seemed to be that there was a mismatch between that dropout rate and the neural loss in the early stages of diabetic retinopathy. Am I correct on that?
Yes, and so the mismatch is a spatial, is a local mismatch, because the perceived wisdom is of how vascular damage affects the retina is that you lose these vessels, and then because there's no longer any perfusion there, the cells surrounding the vessel, they die. And you see that occasionally if there's a big vessel obstructed in the retina, you can get a whole section of the retina will die off, right? It's a kind of like a stroke, the equivalent to a stroke in the retina, and you see that.
Sure.
However, what we tried to establish then is whether we can actually see evidence of that in the tissue. So we looked at where are there non-perfused capillaries, so capillaries without endothelial cells, and then we measured the density of retinal cells surrounding it. And we saw no difference, whether there was an intact vessel or a ghost vessel. So we could not see any difference locally, and that's what we call a mismatch. However, overall, just diffuse across the entire retina, there was a subtle reduction of the count of cells. So there was a subtle loss of neural cells. And that's what we consider the mismatch, right? So whilst you have localized perfusion dropouts, and they can be really, really local, and that has been driving the narrative in diabetic retinopathy research for decades, this local ischemia that drives a whole bunch of complications and pathologies, that is the main problem. But it seemed like from our study that there is actually something more general going on across the entire retina. We obviously said that the limitation is here. We cannot rule out there is maybe vessels that are not perfused, but simply because there is some other pathology of the regulation of the perfusion of the blood vessels, that although the vessels are intact, they may be too constricted to allow blood flow. So that we can't exclude that there is a functional impairment of vascular perfusion independent of a structural impairment of the vasculature. So that's the limitation here. But it seemed like that at least on a structural level, there was no correlation between the local loss of blood vessels and the loss of neurons in the surrounding tissue.
And I was hoping you could touch very briefly on the inner nuclear layer cells, right? Because you saw a pan-retinal loss in that population. Is that correct?
Yes, and that may be that these cells that sit in the inner nucleus may be the earliest ones that are affected. And, it happens that although this was the main narrative, this whole discussion about, what comes first, is the blood vessel damage first that then causes the neuronal damage, or is there also some evidence that there's neuronal damage occurring before the blood vessel damage? And it is a chicken and egg question, which probably will never be resolved, because these are very, very tightly intertwined structures where it's going to be very difficult to separate what happens first. But nevertheless, there seems to be evidence that now so maybe the cells that sit in this inner layer of the retina, that includes glial cells such as Muller cells, but also other cells that are important for the signal that comes from the photoreceptors and up to the retinal ganglion cells, that they may be affected. And there is some evidence in humans who do not have yet any visible, measurable vascular damage, at least according to the traditional methods. They have functional deficits in the retina where you can measure that. The response to colors and so on, to flickers, the electrophysiological response is not quite the same as in a healthy person. So there is some evidence that there is damage within the neurons that may occur before you actually see structural damage to the vessels.
Got it.
So where do you hope to take this work next?
The big question in diabetic retinopathy is still what really drives this pathology, because we still, even though for many decades, this narrative that hyperglycemia is causing damage to the retina has existed, it is not the full explanation. Whilst we know that good glycemic control certainly reduces the risk of developing diabetic retinopathy significantly, from a statistical point of view, the hyperglycemia only can explain a fraction of the total disease occurrence. So there must be other factors which we do not understand, and they are significant. So there's some figures say that it's less than 20% that is explained by the hyperglycemia or poor glycemic control. And the rest, we don't understand why that happens. So there are factors that still contribute to this pathobiological mechanism that drives ultimately the retina down this path of self-destruction. And that we don't understand. And what we're hoping to do is to learn more by looking at the gene regulation, again, in post-mortem tissue from diabetic donors in the retina. So looking at the transcriptional changes that occur within there. So the study which we've been talking about has just looked at the histology And what's ongoing in the lab at the moment is the attempt to study transcriptome within the retina, which is challenging because you have to study mRNA in this instance, which is not as stable as extracellular matrix. So it's more difficult, but that's what we're aiming to go to next.
Very exciting. We'll look forward to hearing more about this work. So, Dr. Fruttiger, we'd like to thank you for joining us today and sharing your expertise and discussing your recent work on microvascular damage and neurodegeneration in early diabetic retinopathy. I encourage the listeners to read the full article in the November issue of Diabetes. Thank you.
Welcome to another edition of Sweet Talk. I'm Kevin Williams.
I'm Darlene Sandoval.
And I'm Kirk Hobegger.
This episode is in honor of World Diabetes Awareness Day, which is November 14th, and National Diabetes Month in the U.S. Today, we're joined by a very special guest, Dr. Giles Yeo, a geneticist and obesity researcher from the University of Cambridge. Many of you might recognize Dr. Yeo from his work on the genetics of obesity and his efforts in science communication through TV, books, and public speaking. Dr. Yeo's research has expanded our understanding of how genetic variations contribute to obesity, appetite control, and metabolic health. Welcome, Dr. Yeo, and thank you for joining us.
Thank you so much for having me on your platform. What's your platform called again? Sweet Talk. Is that what it's called?
Yep, Sweet Talk is a segment in our Diabetes Bio podcast.
Fantastic. I love some Sweet Talk. I know. Thank you so much for having me on your show.
Yeah, we're glad to have you. We always like to start our episode off with a bit of an icebreaker question. You have such a fascinating career across research and science communication, but if you were not a geneticist studying obesity and metabolism, what other field or profession do you think you might have pursued?
Wait. I think I know the answer.
Tell me, Kirk.
Oh, well, I've seen you perform at least 5 times. So I'm guessing that this would be a singer of a rock band. Tell me I'm wrong.
You're not wrong, but what you are wrong is actually the genre of music that I would be singing. So I would want to be a singer if I wasn't doing this business. I would be singing, I don't even judge me, country western. That's what I would sing.
Oh,
The
wow.
problem is country western singing, bald Chinese people singing country western songs in Cambridge. No demand. Can I say that? No demand. So I do disco funk instead. So no, that's what I would do. The other thing I possibly would do, and I tell this to my wife, and she says that I'm a crazy SOB, is possibly actually run a really bougie cafe somewhere, selling some really good gastro cafe, like some really simple food, but with a killer wine list. That's plan B.
Nice.
Nice.
All right. So
Nice.
for the record, I was wrong. And that's why we do the research, right?
That's right.
All right, well, let's jump in. So, first of all, why don't you give us an overview of your perspective on the interaction or balance between genetics and the environment in the context of metabolic health?
Oddly enough, there is a number to this. I mean, it's quite a big range, but because, geneticists have very often used twin studies, identical twins, non-identical twins. So 100% of genes versus 50% of genes in order to try and understand human traits and behavior. And so all human traits and behavior, as it turns out, has a genetic influence. The trick is what is the role of the environment? And that has always been very, very tricky to measure because while your genes are static, the environment is volatile and changes from every single breath you take till the day you don't breathe. And when you actually use twin studies for looking at the heritability of fat mass, which is what we're talking about here, there's quite a big range, actually. It ranges between 40 and 70%. So 40 and 70% of the heritability of fat mass is genetic, which you can then read the yin to the yang to that, is that 30 to 60% of it is down to the environment.
Awesome.
Many of our listeners, aside from our family, right, are researchers and working directly on diabetes. I'd love for you to talk a little bit about the genetic overlap between obesity and type 2 diabetes and how advances in genetic research have helped us understand or better understand the mechanisms underlying diabetes.
That's a pretty tricky question. Let me answer in a different way. So 85% of people with type 2 diabetes have obesity, right?
Right.
But that is not a genetic overlap, interestingly enough, right? So there are going to be genetic drivers that increase your risk for obesity, and hence, because of this powerful overlap, increase your risk of type 2 diabetes. But if you actually parse out the genetic drivers for obesity versus the genetic drivers for type 2 diabetes, then the genetic drivers for obesity tend to involve the brain and feeding behavior. Okay, so appetite. Whereas genetic drivers for type 2 diabetes almost entirely exist within the pancreas and their ability to actually secrete insulin. So I think it depends what we're talking about here. So undoubtedly, stuff that increases your risk for obesity will increase your risk for type 2 diabetes. But if you actually independently take a look at what's driving what, then for type 2 diabetes, it's actually a failure of your pancreas to be able to adapt and secrete enough insulin to sort of surpass the insulin resistance that's actually being seen in the peripheral tissues.
Right. I guess as a follow-up, why aren't the genetic drivers of insulin resistance more evident genetically as compared to the drivers for pancreatic dysfunction?
The answer is we don't know.
Okay.
But I guess
All right.
I have an opinion about this.
Yeah.
I think it's probably one of two things. I would say it's probably more difficult to measure insulin resistance per se than it is to actually measure secretion of insulin. I don't know. I don't know if that's a real reason. It's sort of like the same reason why people say, well, hang on a second. We know that food intake and energy expenditure both play a role in obesity. Why are all the genes to do with food intake and nothing to do with energy expenditure?
Right.
I think there are going to be genes to be found within insulin resistance. They just haven't been found yet. And that therefore the big signal actually comes from insulin secretion. But it's I think it's a really good question. You got the rare conditions of severe insulin resistance that will impact upon the peripheral tissues downstream of insulin signaling, et cetera, et cetera. But from the big GWAS studies that have been done, the big population-based genetics, no signal from insulin resistance at all. And that's a curiosity.
Yeah. Thanks for that.
Yeah, it's really fascinating. And as we're observing World Diabetes Awareness Day and National Diabetes Month, I think it's an important aspect to reflect on the impact of diabetes and obesity around the world. I was wondering if you could speak to maybe a broader global perspective of diabetes and obesity, how these conditions might differ across countries, what unique challenges might exist in addressing them in different parts of the world.
First off, I think that it is a global problem, and I think it respects no borders. It doesn't appear to respect any particular ethnicity either. The differences that we see initially, that we can see, are that the prevalence of type 2 diabetes and obesity in North America and Western Europe and a lot of the high-income countries, I think, are well rehearsed, right? I think we see this. And there are a lot of people with type 2 diabetes and obesity. And hence the increase that is being forecast over the next, 20 or 30 years, there is going to be an increase, but it's not so much compared to if you actually go to a lot of the emerging economies that are coming out, or not that emerging. If we look at China and India, where after all, a quarter of the population of the world live, the rises there are absolutely forecasted to be enormous. And that's because while there are, 300 million or so mainland Chinese people and 300 million South Asian, shall we say, with the same kind of expendable income as us, we go to same places on vacation and eat in the same kind of restaurants. You have to remember that there are about 1 billion people still stuck on the sticks. In China, another 1 billion people in South Asia stuck on the sticks, where at the moment being stuck in poverty means not having enough food, and so therefore being skinnier. Unlike North America, unlike Western Europe, higher income countries, where actually being poorer is associated with higher risk of both obesity and type 2 diabetes. So now, if suddenly you take an extra couple of billion people, okay, and then expose them to exactly the same kind of food environment that we actually live in, suddenly we reach the maximum of our problem. So I think that's number one. We're nowhere near the maximum of the problem, and this is terrifying given how big the problem actually is today. So that, I think, is some kind of forecast. Now, with regards to taking the opportunity to study the diseases in different countries, now we got some really quite interesting differences. Okay, there are different body shapes clearly that actually exist, but there are also two things: there are different BMIs. Different ethnicities actually get risk for type 2 diabetes. So famously, South Asian people, East Asian people that look like me, we don't have to get that large before we tilt into risk for type 2 diabetes and other metabolic diseases, for example. All right. Whereas if you actually look at, I don't know, white people, you go to Polynesia, you look at Polynesians. So suddenly you get to become far larger before you tilt into disease. And so that's the other thing. I think there is a lot of interest now in trying to study the differential metabolic sequelae that comes from gaining weight in different ethnicities. And this, I think, tells us a lot about what I would like to think the underlying causes of type 2 diabetes and metabolic diseases. So, how much fat can we store safely before we actually end up with metabolic disease?
Maybe as a follow-up there, have there been enough genetic variant studies looking at the Southeast Asian populations compared to European, North Americans to try and delineate the genetic differences of what's causing that sensitivity to increase in type 2 diabetes or insulin resistance?
So at the large population level, nowhere close to being enough. I mean, there are smaller, distinct, populations in which people go and actually study individual consanguineous families in Pakistan or Turkish people, for example. But those are actually very, very small studies in which they're looking for rare causes of disease. If you look from a big population genetics basis to try and understand disease, nowhere close. I think it's getting better. Okay, so currently I think the numbers are 85% of the open access genetic information in terms of population levels that are available for anyone to log on and data mine. 85% emerge from white European Caucasian based studies in terms of the individuals. This is better, right? Because 10 years ago it was 95%. So clearly we are getting better. Of the 15% that is non-white European Caucasian, the Han Chinese are building. There's a lot of obviously money and science in China are beginning to build up resources in terms of genetic resources there. South Asians are beginning to sort of claw in. If, however, you look at other populations like Black Africans, oh my God, less than 1%. So less than 1% of the open access genetic information that's available come from Black Africans. So look, we got to do better, because clearly we have to be able to help all humankind. We've got to study all humankind, all of the pathways. I know you guys know this. The biological pathways are universal. The genetic perturbations and influences are going to be private to specific ethnic groups, but all of this information give us more understanding to how these pathways work.
Another really, I think, really cool impact of the work that you have done has been on you being a vocal advocate for fighting weight bias.
Mm-hmm.
And unfortunately, I think this is particularly a problem in the States, but this is a problem that's pervasive in both healthcare as well as society, right? And so can you talk a little bit about why you think this is important, and especially in the context of treatment of obesity and diabetes?
So I think the medical community, and look, we have to admit it, it's part of the problem of weight stigma. I think weight is a very interesting thing, right? And a big part of the problem about looking at weight is that it's a very public thing. So you can see someone, and anyone, you don't have to be particularly well qualified to point out someone with obesity. Also, it's very easy to link their behavior to their condition of obesity. If you go to the grocery store, you can see exactly what people are buying. If you go to a restaurant, you can see exactly what people are eating. And because each of us can only really consider, well, am I hungry or am I not? Do I want the pizza or do I not? Right? And it's a very personal thing in ourselves, but humans are primitive mammals. And so what we do is we model the world based on our own experience. So what we are saying are people who are larger than us making different feeding choices to us. All right. And so thinking, God, how come he needs to have 4 slices of pizza? I just need 2 slices of pizza. And I think that's part of the problem. And this also comes down to doctors, healthcare professionals who are also primitive mammals, and hence make that judgment. And so the reason why I think it's important to tackle weight stigma is because, look, we are failing, desperately failing in order to try and prevent obesity. Clearly, treatment is getting better because of the classes of drugs that are available, but are they made available to the right people? And so until we accept the fact that we can hold 2 thoughts in our head, that obesity is a problem for any number of reasons that you guys know, and not to blame the people suffering from the condition, then we won't be able to treat the problem, prevent and actually treat the problem of obesity properly. So that's why I go and talk about it. Aside from the fact that it's not nice, Okay, leaving the
Yeah.
fact that it's not nice
Yeah.
aside, it is counterproductive, and still costing us a lot of money. So that's why I go out and talk about weight stigma all the time.
It is interesting to see how the new drugs have highlighted to some extent the bias, right? We see it in the lay literature and the headlines, and so layer that on top of this discussion.
Okay, let me take it from a UK perspective. I'm clearly work here in the UK, and last week, our health secretary, we got a relatively new government. I know you guys are about to vote for a new one, but we have a relatively new government, and our health secretary announced we have a national health service in the United Kingdom that they were going to try and target prescription of GLP-1 based agonists, drug treatments, to out of work people with obesity to try and get them back to work. Now, I have problems with this, not because of the use of the drugs for treatment, but because of people saying that, well, you're not gonna in a country with a healthcare system, okay, you are not gonna deserve treatment, okay, Mr. Obese person, until you have some economic value to society. Okay, first of all, what the hell? The secondly, well, what happens if you are an unpaid carer? What happens if you're a parent trying to get your kids to work? I mean, is there no value to society there? So first of all, there's this thing, and then to sort of that adds to the fat lazy person trope. Okay, when we're talking about. Then if you look at the people who are actually taking it, and there is actually a huge amount of shame, and I've seen this, of admitting you're actually on the drug. Now, very many people that I speak to are not, but then other people, I says, you look great or something, right? And so I'm on Ozempic or whatever, right? You sort of, there is almost a shame and to do it. And so that's ridiculous. Who else has been, I feel shameful taking high blood pressure medication. I feel shameful taking insulin for my type 1 diabetes. It's just, I think that talking about obesity is one of the few diseases, and it is a disease that we can talk about in which suddenly you are ashamed if you're taking an actual working drug for it, which is a ridiculous situation to be in. And this is because of weight stigma from the clinician, weight stigma within society, what people expect. Why aren't you doing this naturally? Which is a ridiculous thing to say, but there we go. That's where we're at.
Yeah.
Giles, you've been someone on the forefront of genetic components of obesity and diabetes for a while, and we've touched a little bit on the ethnic or lack of ethnic diversity, right? But aside from that, what most excites you about the future of genetic research in metabolic health, and what should the next generation of scientists and researchers be focusing on?
I think, to my mind, and this is once again, I'm not being prescriptive about this, and people may have other opinions. I think the most exciting thing has happened really over the past 5 years, and that has been the ability to do whole exome sequencing or whole genome sequencing, large sequencing at scale. And the reason why is prior to probably the last 5 years, well, first of all, 20 years ago, we had to do candidate gene approaches, which found
Right.
us only a few things. Then we had GWAS. The problem with GWASs is they highlighted non-coding regions of genes, and trying to link biology to that has been difficult. I have spent much of my life working on FTO, 17, 18 years of my life. It's a very sad story. PTSD still. Don't want to talk about it anymore. Whereas what has happened with this whole exome sequencing at scale is now we're beginning to identify rare changes, coding changes in genes, now population wide, which means that first of all, the gene self-identifies because it's got a mutation in it. Okay. And that means that we can throw biology at it. We can now say, wait a minute here. And so actually the number of new rare causes of obesity, but at a population level that have been identified, but therefore highlighting pathways has been enormous. So that is really exciting because now we can begin to not just see what the known knowns are or unknown knowns are, whatever it is, but we can actually identify the unknown unknowns. And I think over the next few years, as larger and larger populations get sequenced and we begin to parse out, very, very specific traits, I think we're going to have a tsunami of new genetic information and new biology.
Great.
That's a really exciting future there. And, you've done a lot to make research accessible to the public. How important is it for scientists to communicate their findings to the general public? What tips do you have for scientists looking to engage more effectively with a broader audience?
Well, I think it's absolutely crucial. I don't think it should be something, the communication to the broader public. I mean, for a number of different reasons. First of all, most of us, particularly we work within an academic setting, most of us are paid by 99.99% of the population through some way or other. And so they deserve to be told what we do. I think we do have a duty of things. That's the first thing. The second thing actually is, what has the last few years of COVID shown us? What COVID has shown us is that we've seen some wonderful examples of science communication, and we've also seen some absolute god awful examples of science communication. And so what COVID has highlighted, okay, is that bad science communication kills. Okay. Now, within the context of a global pandemic, true. But if you take a step back and you begin to actually think about things, bad science communication will kill in almost any field of science we're talking about, just at different rhythms, at a different temporal scale. Bad science communication in diabetes and obesity will be killing people if we're not taking the drugs, if we're not changing the environment. And so ultimately, I think that no one has to do crazy things like me, okay, going out and do, but everyone can do a little bit of something. You can go to your kid's school and talk about obesity or diabetes or genes. You can write in your local village or neighborhood magazine about that. You can talk, you can appear on the media, on radio, on podcasts. Everyone can do a little bit of something. So the tip would be actually to do something you feel comfortable with and are okay. Sometimes we've got to do things we're uncomfortable with. I think you want to play to your strengths. So me, obviously, I'm not a shy person, and I can flap my lips. And so yes, this tends to be my way of doing things. I tend to talk. But maybe you're a musician. Maybe you have a voice, like an angel. Maybe you write like Shakespeare. Maybe you can draw beautifully. Whatever. There's going to be many, many ways to communicate science, particularly about diabetes and obesity out there. Play to your strengths and do that. And you can do a little bit. You can do a lot. I think all of us have to do something.
And they can always join us here on Diabetes Bio to communicate their science.
To sweet talk people.
Yes, sweet talk. Thank you, Dr. Yeo, for joining us today. And thanks to our listeners for tuning in to this episode of Sweet Talk, which is in honor of World Diabetes Awareness Day and National Diabetes Month. Dr. Yeo, please tell the listeners what you have coming up, or maybe where they can find you and learn more about your work.
They can find me at Giles Yeo, GilesYeo. I've got no weird fancy things, and you do that will track my Insta and various socials down. And I tend to overshare on the socials, and so you'll find out what's happening there.
Any musical engagements coming up?
Dusseldorf next week. I know.
Oh, wow.
Woo! I know. Don't. That's where I'm playing next.
Well, if anybody's in town, look it up. And you have time now, I guess, to book a flight and get there. So that's great. We highly recommend checking out more of what Dr. Yeo is doing. And as always, please like, follow, and subscribe for more Diabetes Bio content.
All right, everyone. Don't forget about ADA's special offer for new and renewing members. That's a 15% discount on a 1 or 2 year membership. Go to professional.diabetes.org/membership, create your account, fill out the application, and use the discount code FALL, all capital letters, 24 at checkout. Act fast because this offer expires November 30th, 2024. Don't miss out on this fantastic opportunity to expand your commitment in the fight to end diabetes by joining the American Diabetes Association.
Thanks to all of you for joining us on the Diabetes Bio podcast. We hope you are enjoying our discussion of the work that the Diabetes Journal is publishing each month. The feature articles we discuss in each episode are freely accessible at diabetesjournals.org/diabetes and are also listed in the episode notes for your reference. As with any podcast, it really helps if you like, rate, follow, and subscribe to the podcast. And if you have any feedback for us, we'd love to hear that as well. We look forward to presenting to you next month.